Hi AP Bio students. I know that cellular respiration can be a tricky topic, so I wanted to make a little video to help you review Chapter 9 in a more visual way and explaining each of the steps. So before we get into cellular respiration itself, I wanted to define some key terms that we talked about early in the chapter. And so feel free to follow along, maybe grab a paper and a pen, and also just different colored pens because we're gonna do some color coding in this little activity. So we open up the chapter with some key terms. We're gonna learn about anaerobic and aerobic. So I'm gonna write this down. So respiration can either happen as an aerobic process or an anaerobic process. Now aerobic, I want you to think like aerobic exercise. You need to breathe a lot, you need oxygen. So aerobic is oxygen. Okay, it requires oxygen, and anaerobic does not require oxygen. And so when we're talking about cellular respiration specifically, we're talking about an aerobic process. So cellular respiration is aerobic, and an anaerobic process is fermentation. Now, some other key concepts early in Chapter 9 was the whole idea of a redox reaction. So I'm gonna write that down. Redox reaction. In other words, it is oxidation and a reduction. Now, in a redox reaction, we are really tracking the movement of electrons. So I like to think of Leo the lion goes grr. So Leo and grr. Leo again stands for loss of an electron, is an oxidation, and grr is gain of electron, is a reduction. So let's see if we can apply this to cellular respiration and talk about the formula of cellular respiration. So if you remember, I like to think about what do we need to survive? Well, we need to eat, we need to breathe. And so the two things that we need in cellular respiration, the reactants would be glucose. You can also say it is C6H12O6 plus 6 molecules of oxygen are going to give you 6 molecules of CO2 plus 6 molecules of water, and of course, ATP. Now, when we think about a redox reaction, what I'm gonna do is I'm actually gonna color code over here. Let's just say oxidation. I'm gonna highlight in yellow, and I'm gonna say that reduction is green. So in oxidation, when you're losing electrons, we can also track how electrons are moving by looking at hydrogen. So you can think movement of electron, track hydrogen. So over here you notice that glucose starts with hydrogens, but then it gets converted to CO2, which does not have hydrogens, and so it is losing hydrogen, is losing electrons, and this is an oxidation. So I'm going to highlight that. This process here is an oxidation. Over here we have oxygen, and it's getting converted to water, and as you can see, it starts with no hydrogens, and then now it has hydrogens. So that means we're actually gaining electrons. And so this is a reduction, a gain of electrons. So then we go into the issue of a reducing agent and an oxidizing agent. Well, it's a little backwards. What's being oxidized is actually the reducing agent, and what's being reduced is the oxidizing agent, okay? You can also think about it as what's being oxidized is causing the other thing to be reduced, and therefore it is the reducing agent. In this case, it would be glucose is a reducing agent. It's causing oxygen to be reduced and gain electrons. And oxygen is the oxidizing agent. It is causing glucose to be oxidized and to lose electrons. So now we have these key concepts down. under our belt, we can now officially start talking about cellular respiration. So cellular respiration has 3 main steps, so we'll talk about each of them. Let's go and start with step number 1, which is glycolysis. If you look at the word glycolysis, it kind of tells you what's going on. Okay, so over here we have glyco and we have a lysis. So I like to think glyco is referring to glucose and lysis is referring to something getting cut. So glucose, we start with 6 carbons in glucose. Here's our glucose molecule. And we're cutting it in half. And so we end with our 2 3-carbon molecules, and that's known as pyruvate. Again, we have 2 of them. We also, in this process, make some ATP. Make 2 of those. And we also end up with 2 NADH. Now let's talk about this ATP. This ATP is not just made from nothing. We actually have an investment phase where we invest 2 ATP in glycolysis. And we have a payoff phase where we make 4 ATP. And that's the payoff. It's also important to note that this ATP is made through a process called substrate level phosphorylation. So in this process, what we have is, let's say that here is an enzyme. Okay, and we have a substrate here that actually has a little phosphate group. And here's ADP. You can see there's 2 phosphates. That enzyme is going to help bring this phosphate group and attach it to the end of the phosphate, thus making ATP. So it's substrate level because here's our substrate. Substrate is right here, and that's where we're getting our phosphate from. So that's glycolysis. Okay, we go from glucose, we're cutting it in half, we're making pyruvate, we're also going to make some ATP and NADH. Our next step is going to be something known as pyruvate oxidation, or I also like to call it the intermediate stage. So I'm going to call it step 1.5, the intermediate stage. Again, also known as pyruvate oxidation. So pyruvate can't go directly to the citric acid cycle. It has to be converted to something known as acetyl-CoA first. So I'm going to go ahead and draw this for you. Here's our pyruvate. It has 3 carbons each. Now what's going to happen is we're going to take off one of those carbons and replace it with something called acetyl-CoA. Okay. So this is 2-acetyl-CoA, and here's your coenzyme A. Now, when you cut off a carbon group, or not a carbon group, a carbon atom, you can think of each of these carbon atoms as becoming a CO2. So we're cutting off 2 carbons here, and so we're going to make and release 2 CO2. In addition, this process helps us make some more of those electron carriers, which is NADH. We make 2 of them in the intermediate stage. Now that we have 2-acetyl-CoA, we are ready to go to our next stage, which is the citric acid cycle. All right, so again, we're starting with our 2-acetyl-CoA. Here it is. Here's one. Here's another one. And what we're going to do is we're going to cut off all of these carbons. Okay, you're going to process it so all those carbons will make us 4 carbon dioxides. And we're also going to make some more exciting things here. So we're going to make 6 NADH. We're also going to make another carrier that's known as FADH2, and we're making 2 of those. two FADH2s. And then finally, we are making some ATP. Oops, let's make it red. So 2 molecules of ATP. Now again, this is made through substrate level phosphorylation. At this point in time, we are ready to move into our last stage of cellular respiration, which is oxidative phosphorylation, stage 3. So this is where the electron carriers play a very important role. They're going to bring those electrons to something known as the electron transport chain. And so what I'm going to draw for you here are some proteins. There's 4 of them. Okay, and these proteins are embedded in the inner membrane of the mitochondria. So there they are. And at the ends, there's something known as the ATP synthase. So I'm going to label this over here. This is the electron transport chain. And then over here we have our ATP synthase, which I'm sure you can guess what it makes. All right, so let's do a little count, okay? At this point, if you count it all up, we have 10 NADH, right? So 2 of them are from glycolysis. We have 2 from the intermediate stage, pyruvate oxidation. And we have 6 from the citric acid cycle. We also have 2 FADH2s, and those are from the citric acid cycle. So what they're going to do is they're going to drop off their electrons. Okay, they're going to drop off these electrons to these proteins, and the electrons are going to fall from one protein and be transported, hence electron transport chain, from one protein to the next. So at the very end, when those electrons reach that last protein, they don't just stop there. In fact, what happens is that they are accepted by something that's very, very electronegative, which is oxygen. Now, that brings up an important point. What's causing these electrons to move from one protein to the next is an increase in electronegativity. So for example, protein number 2 is more electronegative than protein number 1. Protein number 3 is even more electronegative than protein 1 or 2. Protein 4 is the most electronegative of those proteins, and oxygen, as we know, is super electronegative. And so what's going to happen is it's going to attract those electrons very strongly. And so oxygen is going to combine with those electrons. It's going to collect actually 2 electrons at once, add some hydrogens. Okay, add 2 hydrogen ions, kind of just floating around in our matrix. And what that's going to give us is H2O. And so that's why water is one of the products of cellular respiration. It's from this last stage, this oxidative phosphorylation stage, specifically the electron transport chain. Now, as these electrons are going and moving from one protein to the next, what's happening is they are pumping across this membrane hydrogen ions. So hydrogen ions are found inside this mitochondrial matrix, and now they're getting pumped out to the intermembrane space. So if I just label this diagram, okay, right now this is the matrix of the mitochondria. These proteins are found in the inner membrane. Let's see where I should write that. Right here. Okay, and these hydrogen ions are getting pumped out into the intermembrane space. Now what this creates for us is an electrochemical gradient, and it's also known as the proton motive force because, well, hydrogen ions or protons. And because it's an electrochemical gradient and there's a lot of hydrogen ions outside in the intramembrane space, what are they going to want to do? They're going to want to come back in. And the way they go back in is through the ATP synthase. So as these hydrogen ions are floating back through the ATP synthase, the synthase kind of churns and turns, and every turn of the ATP synthase is going to convert ADP plus inorganic phosphate to ATP. And so that is how oxidative phosphorylation makes a lot of ATP. Now your next question might be specifically how many ATP molecules are made in oxidative phosphorylation. Let's look at our NADH and our FADH2 molecules. So we have 10 NADH molecules and we have, or I guess electron carriers, sorry, and 2 FADH2 electron carriers. And turns out that each NADH, okay, so we have 10 NADH, each NADH can make about 2.5 ATP, okay? For FADH2, well, again, we have 2 of them at the end. Each FADH2 can make, well, this is from our textbook, but it says about 1.5 ATP. Now, why does FADH2 make less? Look at where they drop off their electrons. NADH drops off at the first protein, and so those electrons travel and fall down and are able to release energy a little bit slower. But the FADH2 electron carriers drop it off at the second protein, and so they don't travel through as many proteins. Their fall is not broken by as many proteins, and so they can't release as much energy or pump across as many hydrogen ions. And so if we count this up, okay, that means that we make about maximum 25 ATPs from NADH, and we can make maximum 3 ATPs from FADH2. So from oxidative phosphorylation, we can make up to 28 ATP molecules that our body can use for energy. If we now sum it up, oxidative phosphorylation makes 28 ATP molecules. Glycolysis can make 2 ATP molecules, and then the citric acid cycle can make 2 ATP. And so in sum, we can make up to 32 ATP molecules per glucose. Now, of course, sometimes the whole system is not as efficient, and so we'll make, between 30 to 32, maybe sometimes less, depending on how efficient our system is. So I hope this was helpful in explaining cellular respiration, each of the 3 steps, and helping you visualize. Hopefully you're able to draw along, and that it makes a lot more sense to you now. Thanks for tuning in. Bye.